Thanks again to the organisers for this asking me to this wonderful conference. I'm going to talk a little bit about experience I've had with developing a new type of scaffold patch, which is designed to enhance healing through promoting endogenous repair response and the journey from laboratory to clinic. And really this, I suppose, is encapsulated here in this slide, which is the identification of a clinical problem, which I think we all realize we have to some extent, in that a significant number of our rotator cuff repairs are not healing. It would be better to have a healed repair. We need to understand the mechanism of that failure, what is the problem, develop new therapeutic solutions, and then take them through preclinical testing and then human clinical trials. So that's the cycle of innovation, if you like. So the first thing I began to do about 10, 12 years ago now is try and understand, if I could, what was going on in this tendon tissue. It seemed to me that we didn't have as much information as perhaps we should have about the actual biology of the tendon. We could see macroscopically that these tendons are far from normal, but maybe there was more insight that we could gain by actually looking at tissues. So I began by biopsying patients in the operating room and then developed that into the outpatient setting. So using the kind of true-cut biopsy needle, spring-loaded biopsy needle you can see there, I developed a technique where under ultrasound guidance using local anesthetic, we could collect tissue from patients after treatment. So we were therefore able to develop a series of cohorts of patients who present with tendon disease. They have a biopsy at baseline, and then we can repeat biopsy of those patients and follow those patients to see, does the tendon heal, does the tendon not heal, does the patient become pain-free, does the patient remain painful? And we now have a biobank of around 750 tendon biopsies from this cohort of patients. What that cohort has allowed us to understand better is that this is not one disease, it's a complex pathway of disease. The early stages of tendon disease are different to the late stages. The dogma at one time was that this was purely a degenerative process. It's not. It's an exquisitely inflammatory process where there are a mixture of stromal tendon cells and myeloid and lymphoid cells. The myeloid lymphoid cells are relatively small in terms of the proportion. They're about 5%, but they are the orchestrators of what's going on in the tendon. And that is particularly so in the earlier stages of the disease. Pre-tear, tendinopathy and partial tear, there is an intense cellular process going on. As the disease progresses, we see more and more changes within the extracellular matrix. So in large and massive tears, there's deposition of crystals, amyloid, and hydroxyapatite. The extracellular matrix is almost certainly irreversibly changed in late-stage disease, but in earlier stage disease, large, medium, and small tears, there is the potential to alter these processes if we understand the biological processes more. So this slide here just encapsulates, so this is work we've published in a variety of different places, Science Translational Medicine, British Journal of Sports Medicine, and various other journals. But essentially what we think happens is that there is some sort of insult in the tendon. The tendon responds with an inflammatory response. Leukocytes, monocytes, macrophages are involved. In younger people, that results in a repair. As we get older, that response fails to resolve. So we have persistent inflammation. And that's the fundamental problem in rotator cuff disease. Failure to resolve inflammation, resulting in structural changes and structural abnormality. Whether the inflammation persists or not at a particular level is the explanation for pain. Because only if you have a certain level of inflammation do you trigger your nociceptors and have pain. So irrespective of the structural change, it's the inflammatory process that is causing the patient's symptoms, because it's the inflammatory process which is driving the nociception. Once you've had a phase of inflammation, your tendon remains primed. So even in the resolved cases, even when there's a repair, even when the pain is gone, the resident tendon cells are permanently changed. So that's another very important finding. So here from the mechanism, in very brief summary, inflammation is crucially important to understanding tendon disease. Immune cells are the regulators of inflammation. They change the tendon population. And that tendon population is not uniform. There are different subtypes of tendinocyte within the tendon. Inflammation and pain are linked. Resolution of inflammation is key to taking forward better therapeutics in tendon disease. So what we've been looking at is a series of different options. I'm not going to talk about the pro-resolving mediators, which is one line of attack we're taking. I'm going to talk about bioactive scaffolds, particularly for the management of later disease where we're trying to repair tissue. So what about scaffolds? Well, we've just undertaken a systematic review. There are about 52 studies looking at scaffolds in cuff disease, 4 randomized trials, 11 observational comparative, and 35 observational case series. The findings of these studies are that there does seem to be evidence of some improvement in shoulder function in pain with synthetic patches, but not with extracellular matrix patches. There's a reduced likelihood of scan-confirmed re-tear for synthetic and allograft patches, but not xenograft patches. And there's no difference in complications between the patched and the non-patched. Now, these studies are not brilliant, but that's what's out there. There have been problems with patches, and I'm sure you're all aware of the issues, the inflammatory foreign body response to the Restore patch, causing it to be removed from the market. We've just done some work biopsying patients after cuff repair with patches, and we've looked at standard repair compared to a human allograft compared to a porcine graft. And essentially, we do see a cellular response in patients who have had patches. It's difficult to know whether that's going to be helpful in terms of repair. We haven't done enough of these patients yet. We have unfortunately seen significant foreign body response in some cases of xenografts. So in the Permacore patch, we have some cases of really exaggerated response, which to me causes considerable concern. So what has our program aim been? It's been to transform surgical scaffolds from being simply inert and mechanical support to being resorbable and bioactive, so-called smart materials that assist with healing and modulate inflammation, causing the generation of new tissue. And then after a period of time, and you can modulate that by using different polymers, you get resorption. So this material does not stay forever and cause potential for long-term problems. So what we based the work on, and I began this about 10 years ago, was the basic science identification of the exquisite sensitivity of cells, human cells, to certain types of biophysical cue. If you drop the scale of the material which cells come into contact with to around a micron, so micron or submicron, so-called nanoscale, you begin to change quite dramatically the response of cells. So material architecture, biophysical cues are capable of a physiological driven response. So an example of that is that you can take a rodent heart, you can completely decellularize it, so you leave a shadow heart. You can put stem cells onto that heart, infuse it with a medium, and after a period of time, those cells will differentiate into cardiomyocytes, and the heart starts beating again. So it gives you a sense of the power of biophysical cues in terms of directing cell fate. So the candidate technology we landed on, we tried all sorts of things, 3D printing, various other things. We ended up looking at electrospinning, which is a technique whereby you can draw polymer from a nozzle using an electric charge differential to produce fibers that are micron or submicron. And then by changing the collection plate, you can orientate those fibers essentially to be mimetic or biomimetic of the extracellular matrix of tendon. So you can see in the pictures on the left, a rat native tendon and tendon fascicle, and the pictures on the right, an electrospun sheet and an electrospun filament. So you can essentially produce a mimetic extracellular matrix using this technique, using safe polymers, so polymers used conventionally in surgery. So it's not the polymer that's the issue, it's the design of the fiber. And what that does is it allows you to demonstrate, well, it allows us to demonstrate that by growing cells on an ordinary patch, you get no response. You can see the cells in the scanning electron microscope picture. But on an electrospun patch, these stem cells, within 24 hours, have dramatically changed their shape. What this live cell imaging picture does, this was done by one of my postdocs, Adrian Kendall, in the lab. So these are tendon cells from a patient with a rotator cuff tear. The fibers of the electrospun patch, you can see, are smaller than the cell, and they're going from top left to bottom right. The grid is the dark patches, which the electrospun fibers sit on. If you were to put these tendon cells onto a conventional scaffold, they would just sit there. This shows you what happens over time lapse here, over 24 hours, what happens to these cells as they come into contact with these materials. So you can see a dramatic response. The shells stretch out, but they also move, creating that, as Jess said, that all-important shear effect on the cells. As the cells change their shape and move, they change their behavior. And they do this through an epigenetic process. So this change of shape and this shearing of the cell sends a signal into the nucleus that deacetylates and demethylates the chromatin, switching on genes that have been switched off, and you get production of extracellular matrix proteins. So we are changing the behavior of cells through changing their shape and changing their movement by switching on the cells. So the next thing we had to think about was, how do we take this exciting technology and create something usable as a surgical material? So how do we create a mesh, a suture, and create a tube? And I'm going to talk about the work we've been doing on mesh production. So in order to, these materials are inherently flimsy, so you can't put electrospun materials in on their own. They won't take sutures, they're not surgically usable. So the technology we developed was to laminate, to create a composite of a woven material on one side with strength and integrity, and electrospun on the other side. And this is an SEM of a blown-up picture. So this is 7O polydioxin and PDO on the woven surface, and then these are submicron fibers on the undersurface, the bioactive electrospun there. And you can change the depth and size just by electrospinning for longer. So you can tweak the actual physical characteristics, but the essential thing is that you have two components. On one surface, the bioactive layer, and on one surface, the mechanical layer. So we took this then through a series of stages of, firstly in vitro, I've shown you some of the data from the cell work, and then animal work. This is a rodent model, and this was what Mark Murray did when he came over from the Mayo to work with me, so send my best wishes back. And we found significant tissue ingrowth within weeks into these patches and no evidence of a foreign body response. And then we developed a non-healing, this was some work I'd been doing on stem cell therapy. We wanted to find a non-healing tendon injury in a large animal. It's actually very difficult. Most large animal tendon injuries heal. So we actually end up using the deep digital flexor tendon of the sheep, which is creating a lesion in that. It never heals. And it doesn't heal if you give the sheep stem cells. But when we covered these lesions with our nanofibers, they all healed. And so that was encouraging. We also found, at a histological level, ingrowth of cells from the surrounding tissue and ingrowth of vessels. And here you can see in a graphic form that we're showing, so the growth into the woven material is relatively limited, if not none at all, but the growth into the electrospun layer is dramatically increased. And what was encouraging is it wasn't just the fibroblast tenocytes that were growing into the electrospun layer, it was vessels as well. So they seemed to be drawing vessels into the material. We also looked at whether or not these sorts of responses would be the case in human disease, human tendon disease, and we found there was a marked difference in fibroblast response from healthy tendon to diseased tendon, and we found an increased response of inflammatory cells. So these materials seem to be particularly capable of changing the behavior of diseased tendon cells and of primed inflammatory cells. So just the sort of response that we were looking for, the potential to generate an endogenous response in your tenocytes and to change the behavior of your inflammatory cells. So the summary of this work is that nanofiber materials influence endogenous cell behavior. This response differs between normal cells and disease cells, and nanoscale materials may have a regenerative and immunomodulatory function in human disease. So we might be able to use them for treatment. I've just got a couple more slides, if that's all right, because there is now this crucial issue of how do you take this innovation into clinical practice, into patients, and determine whether or not there's benefit for patients. And I just want to spend a few moments just reflecting on the issue. For me, the exciting thing in orthopedics going forward over the next 2 years is the increasing engagement of what we do with quasi-pharmaceutical responses of our treatments, so the bioactive biological treatments. This is really exciting. I think it's the best way that we're going to change the disease processes. But it's going to require a new approach to evaluation. The materials we're using are going to be quasi-pharmaceutical. We need to be careful about what we do. It's possible to get this wrong. So this is the example of the trachea work that was done by Macchiarini at the Karolinska. It's a fantastic idea. This is a stent. This is a tissue engineering stent, which has stem cells on it and cartilage cells on it. The problem was they did the studies really badly. They were fundamentally dishonest, resulted in the board of the Karolinska being sacked. 3 of them were on the Nobel Prize Committee. We have a responsibility to translate these new technologies effectively. And one of the issues is regulation. This is a quick summary slide of what the regulatory system in the United States has been up to present. And it shows you, in the left-hand side, the cost and time it takes to get a pharmaceutical into market. So it's $500 million and it takes you 10 years. Conventionally, orthopedic implants have been taken through what's called the 510K route, where all you have to do is demonstrate equivalence. No clinical trial, it costs you $5 million and it takes you about 3 years. There is a more sophisticated and complicated route for surgical devices, which was the intention from the outset of this system, which began in 1976, which is pre-market approval. It's $100 million, and it takes 5 to 8 years, and it does require a clinical trial. The question is, how fit for purpose are these in the context of biological types of therapies? Well, this is one of my postdocs decided to have a look at surgical meshes. So we actually found no surgical mesh on the market at all approved by any route other than 510K. This is what happens with equivalence. So this is one mesh, Merceline mesh, on the market in 1976. It has spawned through equivalence 175 other meshes. So they're all on the market on the basis of equivalence. 2 of them have been removed by the companies for safety reasons. They have 12 predicates on the market through equivalence, still on the market and still being used. So there is a problem with equivalence, and it's not better in Europe. Europe, we have the CE marking system, where we have a separation of approval and monitoring, which is conjoined in America with the FDA. Notified bodies approve your product. They give it a CE mark, and then you can market it. So one of my chums in Oxford, who heads up the evidence-based unit, I said, I've got real issues with this. He said, I'll tell you what I'll do, Andy. We'll see what we can get through on CE marking. So he took the bag you buy oranges in at Safeways, and he got it approved on and CE marked through on the basis of equivalence. So he took the mesh, he put a nice picture, he produced all the documentation, and he got it approved as a surgical implant. So, yeah, there are issues. Now, unfortunately, what's happening as a consequence of metal on metal and pelvic meshes and other things, there is, in my view, an overreaction of the regulatory authorities, particularly in Europe, but also in America. And what we're seeing is a shift towards what to me is going to be an over-onerous regulation of surgical innovations, which I think is going to be counterproductive. So what we've decided to do is not commercialize this. we are taking this product through 2 stages of trial, safety and feasibility, and then an efficacy study. These trials will cost about £1 million, so not too expensive. And then we're going to go to the FDA and for CE marking. So we've decided to try and carve out a new way into patients which proves safety and proves efficacy, but isn't over-onerous in terms of cost. I'd just like to thank my team, who you see here, who are clinicians, engineers, biomanufacturing team, lots of regulatory advice. Thanks very much for your attention.